Alright, so this last video where we're talking about just generic nervous system stuff, we're going to talk about neurotransmitters. So we're going to talk about a couple different classes of them and how they function, and talk about examples, talk about agonist versus antagonist, and then different ways we can regulate this, and then different types and where we find them, what they do, right, how we regulate those, and then really just get into a long list. So if you're looking at this, we're going to talk about a whole bunch of different neurotransmitters, just what they are, examples, and function. So first and foremost, we need to talk about neurotransmitters and what they do. But the neurotransmitter is a chemical signal that is going to bind to a receptor. And when they bind to a receptor, their whole job is to basically open an ion channel. Because remember, with these neurotransmitters, we're trying to either excite or we're trying to inhibit. And we can do that by allowing ions in or out, right, through a certain channel. So we have ligand-gated channels, which we've already talked about, where like the neurotransmitter binds to it, and that's going to cause the channel to open, right? So ligand-gated channels are ionotropic receptors, right? These are going to just open the channel when the neurotransmitter binds. So example would be like the nicotinic acetylcholine receptors. Other ones are G protein coupled channels or metabotropic receptors. So this is where the receptor is separate from the protein, right? That serves as the ion channel. So like the receptor and the channel are separate, and that when the neurotransmitter binds to the receptor, we have the G protein that will indirectly open that. So if you remember the G proteins from talking about the hormones with endocrine, it's the same concept, right? In that the receptor and the effector, basically the effector molecule in there, are separated, and the G protein is the go-between between those two. So in this case, we look at examples like the muscarinic acetylcholine receptors, or things like norepinephrine or dopamine receptors. So this is just the picture showing the difference, right? We have the ligand-gated ones. We saw these as one of our examples when talking about the electrical activity kind of stuff, and earlier on with neurons. This one's very straightforward. Neurotransmitter binds to the receptor. The receptor also serves as the channel, and so it opens when the neurotransmitter binds. Simple enough. G protein coupled, again, the receptor and the channel are separate, and the G protein is going to be the go-between, right, between the two. So when the neurotransmitter binds to the receptor, it activates our G protein, which then goes and works with effector proteins to open that channel. Both of them have the same end result, it's just the mechanism for opening it. Ligand-gated is very straightforward, where G protein coupled, we have a couple extra steps in the mix. Now, in terms of regulating these transmitters, right, we have agonists and antagonists. So when we think about a neurotransmitter and the actions that can be modified, we can stimulate or inhibit the neurotransmitter from being made, right? If the neurotransmitter is not there, then it can't do its action. If we need it, we can make it, right? We can block or we can enhance its release. So block the release of the neurotransmitter or push for the neurotransmitter to be released. We can stimulate or inhibit the removal of the neurotransmitter, so stop the removal so the neurotransmitter keeps working, or do remove it and we stop the action of the neurotransmitter. Or we can go right to the receptor site, so block or activate the receptor site. If the receptor is blocked, then the neurotransmitter can't bind even though it's there. A lot of the medications for certain mental health conditions are working by doing one of these things, right? By modifying the actions of a neurotransmitter. Now, agonists are going to stimulate a receptor. So, for example, nicotine for a nicotinic acetylcholine receptor, and muscarine for a muscarinic acetylcholine receptor, whereas an antagonist is going to inhibit. So like atropine will be the antagonist for muscarinic. So these are going to be medications or drugs that Again, these are not neurotransmitters. These are like drugs that can cause these effects, right? So that's important to note. And you'll see like classes of drugs be called agonists or antagonists in some cases. So the neurotransmitter gets released. That's going to allow the presynaptic cell to talk to the postsynaptic cell, but we don't want the neurotransmitter there forever. And so we do need to remove it from that synaptic cleft. And there's a couple things we can do. One, it can just diffuse away, right? It just diffuses away, it just disappears basically, or like spreads out. Or the presynaptic cell can actually take it back up. So it actually pulls the neurotransmitter back up into the presynaptic cell. Or enzymatic degradation. So for example, acetylcholine is broken down using an enzyme called acetylcholinesterase. We talked about this one when we talked about the synaptic cleft. But acetylcholinesterase will inactivate acetylcholine. It'll break it down into choline and acetate. And then the choline, right, these things can be taken back into the cell and reused, rebuilt back into acetylcholine to be released later on. So we have a couple options in terms of removing a neurotransmitter that we've already utilized and we've already sent that signal and we don't need that signal being sent anymore. Now we have classes of neurotransmitters. Some are classed as excitatory, others as inhibitory, and some can actually do both. So it depends on the location. When it's both, it depends on where we find it. Some also function as hormones, so like norepinephrine, and it will function both as a neurotransmitter and as a hormone released from an endocrine gland. So we have some things that are kind of multi-purpose in a way. So here are just some different, just a table showing some different ones. I'm not gonna pause here or anything. You're welcome to pause here. I'm gonna take us through the classes though one by one. So let's go ahead and take a look, and starting with the monoamines. So the monoamines, these are going to be derived from amino acids. So we have catecholamines, we have serotonin and histamine. These are going to be the three. Now, catecholamines are going to be derived from tyrosine, and it's going to include dopamine, norepinephrine, and epinephrine. Serotonin is going to be derived from tryptophan, and histamine is derived from histidine, hence the name. So that's where these ones are coming from, right? These monoamines. Now their job, right, this group, they're going to bind to metabotropic receptors in a postsynaptic cell. When they bind, they're going to activate G protein coupled receptors. So they're going to bind to a G protein coupled receptor. Right, which means they're going to go through all the same steps to activate, right, an ion channel. Right, we saw this same cycle, right? The adenylyl cyclase converting ATP to CAMP then activating kinases, and then kinases going down the line to open up the ion channel. But they're binding to a G protein coupled receptor. That's one of the things I'd like you to take away there. When they get released, they get taken back into the neuron that released them, the presynaptic neuron. And then they get broken down by monoamine oxidase, or MAO. So you may have heard of medications called MAOIs. MAOIs are monoamine oxidase inhibitors. So these are going to be medications that basically block the degradation of monoamine neurotransmitters. So something like serotonin doesn't necessarily get broken down. This prolongs the effects from the monoamines, so this is usually for things like depression. It can be used for panic disorders, anxiety. You do have to be careful with certain over-the-counter things if you take MAOIs, because they can interact. So things like St. John's wort, right? Because tryptophan, so let's say you have like St. John's wort that has a lot of tryptophan, that tryptophan can be used to make serotonin. Serotonin would be one of these monoamines, and so if you are not breaking down the monoamines you're releasing, and then you're also making more, you can end up with way too much, right? So that's the monoamines. If we look at one specific one, we can look at serotonin. Serotonin is used to affect your mood, behavior, and appetite, which is why we have medications that target it when it comes to treating things like depression and anxiety. Serotonin is used by neurons that are found in the RAF nuclei, which is like in the middle of the brainstem. And there's a lot of different receptors that bind with serotonin. Serotonin is used diversely, we'll say. And there are certain medications like SSRIs, or serotonin specific reuptake inhibitors, kind of like an MAOI but more specific, are used to treat things like depression. So like Zoloft and Prozac would be this, but also recreational drugs can promote serotonin as well, so like LSD and whatnot. Another one is dopamine. Dopamine is an excitatory monoamine, and this is used for movement, reward, and addiction. So it helps you with moving, but it also is going to be like the reward system. So when you do something that, like you eat something that tastes really good, you get a dose of dopamine, right? You're like, man, this is fantastic. And so that's what can lead to people getting addicted to things, because they're getting this dopamine release when they use that substance or they do that thing or whatever. The big thing that I'm worried about here, though, is knowing what dopamine is generally for. It's excitatory, it's involved in movement, reward, and addiction. Some other ones, norepinephrine. This is catecholamine or a monoamine that is used both in the central and peripheral nervous system and is excitatory for the most part. So in the peripheral, this is going to help with smooth muscles, cardiac muscles, and glands to promote fight or flight, which we'll talk about more a little bit later. And in the central, we're looking at things like alertness, memory, as well as arousal. So we'll talk about like fight or flight, and we'll look at the things we think about, like, getting ready to like fight or flight, run away, but there's other things besides just the standard, like, I need to fight for my life type response, including things like arousal, like erection and ejaculation, are even going to be controlled by the autonomic system. So norepinephrine is going to be excitatory, right? Generally speaking, talking about the stress response, as well as things like alertness, memory, and arousal. Acetylcholine, this is a cholinergic one, so it's made by cholinergic neurons, and it can be excitatory or inhibitory. It's both. In some parts, it's excitatory, and it's inhibitory in other parts. The thing that you want to keep in mind with this is that it's going to be excitatory when we're looking at nicotinic receptors. It's going to be inhibitory when we're looking at muscarinic receptors. Usually we think acetylcholine, we're going to be thinking of the muscles. It's one of the main neurotransmitters that will allow for muscle contraction, and it's also going to be involved in the autonomic nervous system when we look at the, quote, rest and digest side, the parasympathetic nervous system. So the two types, nicotinic and muscarinic. Nicotinic we find with the skeletal muscles, so at the neuromuscular junction, and then some parts of the central nervous system, as well as the autonomic nervous system ganglia. And then in the muscarinic ones, we find this in like the cardiac muscles, glands, and parts of the central nervous system, and that's inhibitory. The thing that I'm worried about most here is knowing nicotinic is excitatory, muscarinic is inhibitory when it comes to those receptors. So we can inhibit this to some degree. Cholinesterase, if you remember, acetylcholinesterase was the enzyme that breaks down acetylcholine. There are drugs that can inhibit that, right, so that we can increase the amount of acetylcholine that is acting in that synaptic cleft. So this is helpful for things like myasthenia gravis or in urinary retention where someone cannot urinate because the muscle can't relax. And so that would be one application of that. So myasthenia gravis is just an autoimmune disease, right? Antibodies are binding to these nicotinic acetylcholine receptors, which prevents acetylcholine from binding, which means muscle weakness. We really see it like in the face. These medications, though, these are going to block the activity of acetylcholinesterase to help treat the symptoms. Basically, allow acetylcholine to stay there longer so that acetylcholine can bind to receptors when it can to prevent that weakness. Again, I'm not worried about you memorizing a bunch of conditions. I'm more worried about you remembering normal function. We're talking about them only so that we can kind of understand the normal function, in this case, of acetylcholine. Looking at some other classes, we have glutamate. This is an amino acid that is excitatory for things like learning and memory. So right now, hopefully you have lots of glutamate working to help you remember what we're talking about. Glycine is going to be an amino acid, one that is inhibitory, and we need this to help the muscles relax, especially when we're looking at antagonistic pairs of muscles. So something like your biceps and triceps, if your biceps is contracting, you don't want your triceps to contract at the same time. That would be very painful and damaging. So glycine can basically inhibit the triceps muscle from contracting. That way your biceps can contract and your triceps can relax. So glycine will help with that kind of thing. GABA, this is an amino acid, or gamma aminobutyric acid. I'm fine with you calling it GABA. I'll probably just call it GABA on something like a test. I'm not going to spell that out. This is an amino acid that functions as an inhibitory neurotransmitter and is helpful for motor control. We see that when the neurons that make GABA degrade, when someone has Huntington's disease, we see problems there. We see that that's one thing that happens in Huntington's disease. When they bind, it allows chloride ions to get into the neuron. Same thing as glycine, right? They both allow for chloride ions to get in. And then neuropeptides, so these are little short strings of amino acids. They're not quite a protein, but they're also not single amino acids, which is why we give them the name neuropeptides. CCK is going to be what makes you feel full after you eat. Substance P is going to mediate your sensations of pain. Neuropeptide Y is going to reduce stress response, right? Reduce pain, increase your appetite and whatnot. And then we get to the opioids. So you do have what we call endogenous opioids, meaning they're ones you naturally produce. These are inhibitory. Here's this list. I'm not worried about you knowing like a bunch of them, but the thing that we get here is that this is going to mediate reward, a feeling of well-being, euphoria. Sometimes you get this after exercise. So if you've ever heard of runner's high, that is from a release of your natural opioids in your brain. We also do use medications that bind to the same receptors that opioids work on. So things like opium and morphine, heroin, those things. Obviously, some of these are more useful than others. Some are, very much dangerous recreational drugs, whereas others, like morphine, give pain relief, and if used properly, can be extremely beneficial for a patient. That being said, because they give like euphoria and it mediates a reward and a feeling of well-being, you can understand how these can easily become abused and or addictive. Endocannabinoids, these are little fatty acid chains that are excitatory. THC from marijuana actually binds to these same receptors. So you have your natural endocannabinoids, and then of course THC, which is from the marijuana plant. And then nitric oxide, this is a gas produced by the neurons that is excitatory. This is going to diffuse across and basically the autonomic neurons can secrete this in the digestive tract, respiratory system for muscle relaxation. This is what's going to help with things like developing an erection, and it's also how the drug Viagra works. It increases the nitric oxide release, which is why it can help with something like erectile dysfunction. So I know that that was a lot, and I want to remind you, I'm mainly worried about with this knowing the classes and like their basic structure, like endocannabinoids are fatty acid chains, things like that, and then knowing what they do on a basic level. I'm not worried about the medical conditions that people can develop when they have a problem with them. So if you're worried about that, don't worry. I'm not going to ask you which neurotransmitter is involved with myasthenia gravis or anything. I would rather you just focus on what are the neurotransmitter classes and what do they do for the most part. Are they excitatory? Are they inhibitory? And then what body parts are they really impacting? So for example, going back to like here, CCK makes you feel full, or going back to acetylcholine being both excitatory and inhibitory, depending on what you're looking at. In excitatory, we're usually looking at muscle and whatnot, where like skeletal muscle, and muscarinic, we're looking at cardiac muscle, glands, things like that. That's what I'm more worried about. Some of this stuff, like the 2 dopamine systems, I'm not as worried about. I'm okay with you just knowing dopamine being excitatory, that it's a monoamine, and that it's for reward addiction and movement. That's what I'm going to be worried about here. So just keep that in mind as you're going through. There's a lot in this one. Focus on the classes, make your flashcards, do whatever you need to for just talking about the kind of surface level of these different neurotransmitters.